A very good morning to everyone. I, Sanjana A, Assistant Professor, Department of Pharmaceutics from Yenepoya Pharmacy College and Research Center. It gives me an immense pleasure to welcome all the delegates, speakers, colleagues of the constituent colleges, and I am very happy to inform that we have delegates from Karnataka, Kerala, as well as Andhra, Telangana, Tamil Nadu, and Bihar and North and other north sides, and so on. I request all the delegates to please put off your microphone and camera off. And any questions or queries which will be asked in the chat box, and it will be responded at the end of the session by the speaker. Now, the feedback link will be placed in the chat box at the end of a session during the question answering. Now, I request our beloved principal sir to address the gathering.
Good morning, all of you. It gives me a great pleasure to extend you all a very warm welcome on behalf of Yenepoya Pharmacy College, our incubation center, as well as Yenepoya Deemed to University for this webinar on regulatory perspectives in product development. Just to mention that though we have conducted many webinars from our university, purely from Pharmacy College, this is the first webinar we have conducting. And I would like to welcome today's speaker, Dr. Sushil Chaubeji, who has accepted our invitation to be a part of this. And it's happy to mention that the participants are across the country. Just now the Sanjana mentioned about, so they are from the various different states, and many students were able to see, and also the faculty members, and also the personnel belonging to industries. So behalf of the Pharmacy College and behalf of our university, I welcome all of you for this webinar. And definitely the participants will not be disappointed. At the end of the session, definitely you are going to take home a message delivered by our respected speaker, Dr. Sushil Chaubeji. And also, I express my sincere thanks to our speaker that though as a far place he is accepted and taken his time to be a part of our webinar. So since 2 to 3 weeks, we are keep on constantly touching him regarding and requesting him to be a part of this, and he is happily accepted. And today, so we made the session. And I also extend my sincere thanks to the Staff Welfare Committee of the Pharmacy College who have taken pain to arrange this, and also Mr. Asim, the head of the incubation center. So who is the first person to initiate that? So can we have a collaborative work to start this kind of webinars? So with this brief, so anyhow, Dr. Rukia, the organizing secretary of this program, is going to give the brief introduction about this program as well as our speaker. And I appreciate the patient listening of all the participants. Thank you and welcome for the seminar.
Thank you, sir. Now, I request our organizing secretary, Dr. Rukia Sultana, Staff Welfare Committee, to welcome the speaker.
Good morning, on and on. It's my privilege to welcome today's speaker, Dr. Sushil Chaube sir. Dr. Sushil Chaube sir is a medical postgraduate with 17 plus years experience in different fields like pharmaceutical and medical devices, regional and global leadership, R&D at different global MNCs and the big pharmaceutical companies like Novartis, Allergen, Sun Pharma, Torrent, Piramal, etc. At present, Sushil Chaube sir is working as a founder and managing director at Trans Integra Healthcare Private Limited, Bhubaneswar, Odisha. As sir said, that will not be disappointed. Sir will be enlightening us with his talk on regulatory affairs. I welcome you, sir.
Thank you, ma'am. Sir, now it's a session over to you, sir. Sir, please put off your microphone, sir. It's not audible.
Sushil Chaube sir, your microphone is off. Thank you, sir.
Is there's screen away, like the slides can be seen, madam?
Yes, sir. It's visible.
Okay. And is the audibility like clear? The
Yes,
audio?
sir. Yes, sir.
And the slides are visible, right?
Yes, sir. You are very much audible and your slides are clear.
Okay, great. Thank you. So welcome everyone. And the title of the slides might be slightly different from the communication, but that's my mistake. But in essence, the subject matter is same. Basically, like wanted people to be aware of what is the what do we mean by regulations, regulatory and regulations. And it applies, like regulation applies to every field, right? Right from construction to finance to different industries, airline and all R&D related activities. Because we are in healthcare and pharmaceuticals, medical devices or biologicals, here, like I would like to mention, like biologicals, I'm including in pharmaceutical for the convenience. They are quite similar, a little bit different. So because we are in healthcare and human life or sometimes like animal health is involved, particularly like human health. And the concerns about benefit and risk are paramount. So there will be quality systems. Everyone from pharmacy will be aware of the quality systems, the word and the nomenclature. And apart from that, regulations has to come in. You have to set certain laws that has to be followed. And there will be people who will be monitoring, review and monitor and see whether you are compliant to those laws or not in dealing with human health and well-being. And also like internally, like you should have some people who are competent to take you through the guidelines set by under the law and regulations. So briefly, like that is the essence of the presentation today. You can note down if there are questions. Normally, I like to, in a live presentation, take the question during the session. So I don't know whether it's a good idea to take it right in the meeting, but you can take it, note down the questions. And I don't know how much time we have for question and answers. Maybe like 10-15 minutes or more, madam?
Sir,
So,
we have 10 minutes for the discussion.
Okay, right. Thank you. So coming to the very familiar slide, and most people are aware, right? Those who are into pharmacy or healthcare. Some of you may not be like aware of the entire life cycle of a product development in whether it's a pharmaceutical, biological, or medical devices or diagnostics. So there will be, I'll briefly say like a long, hard, unpredictable road of development. And normally like it takes about 8-10 years for a chemical drug, similarly for biologicals. From the time of the first patent filing, it might be about 7 or 8 years. And the timeline in the slide, which is taken from a textbook, it shows 5 years to 10 years. It's an old book, and the timelines have shrunken a little bit. So, and the average time would be about 8 to 10 years. And from the time of the first patent filing, which is after the some proof of concept and some early discovery work related to chemical or synthesis or pre-formulation. Any of the terms that I'm using, and if you are not familiar with those, you can also note it down. I'll try to explain it at the end. So from the patent filing to the market to the approval and market launch, it will be about 7 to 10 years, and total like the time that you get to sell a product within the patent framework would be about anything from 7 years to 10 years or 12 years. The question is like how quickly you can get the different stages of research, discovery, preclinical, the clinical development, different phases of clinical development quickly done, so that you can reach the business, or you can reach society earlier. So some of you may not be like, I have been dealing with a lot of people who come from pharmacy background and life science background. And early days when you take like some basic interviews, interns and others even like some like with advanced certifications, sometimes people have not a complete, I'll say, assessment of what is a preclinical and what is what includes preclinical development. So sometimes like preclinical is animal toxicology, animal studies, pharmacology and toxicology. For us, like it means like even the in vitro studies, all the in vitro studies that you will do becomes part of the preclinical development. And it might be related to cells or in vitro or living organisms in vivo, or it could be chemistry and formulation development, like dissolution, solubility, permeation test, which are physicochemical parameters. This slide shows that there is a huge amount of ideas, the discovery ideas that will not pass through the different stages and might fail or get rejected because of various reasons at different stages of development. So as you see, we call it the funnel. So here it is horizontally placed. Usually like the funnels will be, look, okay, what goes in and what comes out is a very small amount. So here like a few hundred and thousand of drugs, potential drugs, potential targets, potential drugs, potential medical devices would be entering, even the biomarkers now of biomarkers. So thousands and thousands of biomarkers will be getting evaluated, like conceptual stage, which is a research, basic research and discovery stages. And most of them, they will not pass through the discovery stage. They will, some publication work will be there, some will even not get published, some will reach the stage of patents, some will reach some preclinical work along with patent and other things. And full-fledged like pre-formulation and chemical development work, like only after you know that there is a good chance that it might pass through some of the preclinical like in vitro tests, then you try it out, and most of them, they will not succeed. We'll see in the next slide why they don't succeed. So what is the reason for attrition, like failure of drugs or candidates? I'll say candidate products. And this is typical of chemical pharmaceuticals, not for medical devices. So the main cause is like PK, lack of efficacy, animal toxicology, failure in animal safety and toxicological studies related to, later on related to adverse events in human beings. Now, the reason for showing this is like each of the parameters, like how does a product fail or succeed, and who decides it? Is it the research team? Is it the another independent reviewer? We'll see that, whether the name is synonymous with a regulator. So here, the regulators should be familiar with each aspect, right? So that you are able to determine whether a product is meeting the requirement of PK or not. And typical pharmaceutical industry, we say from a commercial point, and as you become a senior guy in development, how early can you kill the product? It is unnecessary wastage of resource, time, money, the priority of focus, everything. So just to give an idea, like in companies as big as Novartis, where there were 18,000 R&D people. So some of our global presentations to senior management, particularly like the commercial people, because they are the guys who needs to be convinced so that they can allocate the resource, the financial resource, which will translate into human resource as well as equipment and space, etc. So sometimes like there are at least 12 or 15 therapeutic areas like oncology, cardiovascular, neurology, etc. And each of them, they would have about 100 products in the R&D pipeline, like very early development, right in the pre-formulations or animal tox or lead optimization, etc. And sometimes people, they used to get 1 minute to present. So within 1 minute, you have to convince or 2 minutes to convince the management whether you are good enough to get filtered to the next step. So sometimes we spend about an hour and so on and to decide. But in a very senior level in the management, I remember like the CEO Joseph Nez and David Epstein and a few others like the oncology head, they will spend 1 minute, not more than that. And the therapeutic area head has to explain within a single slide. And within a minute, he will be told yes or no. And here the therapeutic lead will be taking inputs from everyone, which includes regulatory. We'll come to that later. Now, as a healthcare provider and as a person who is a medical professional and a regulator, I'll say that it's better not to treat than treating with dangerous goods. And long back, I had a colleague from Jadavpur University, a PhD, and he did his this thing in pharmaceutics, MSc and PhD, I think in pharmaceutics. And this used to be Torrent Research Center, like 2005 and 6, 2005, I think. So as a formulator, he will say that if I'm not able to convince myself that I will be able to give this formulation to my kid, and he had a small kid, and he says that very experienced guy, And he says that if I'm not convinced about the quality of my product and that I can give it to my family members, I'll not proceed with the, after the analysis is done, the pre-formulation stage and analysis is done, I'll not proceed further. So similarly for the entire stages like clinical pharmacokinetics or animal talks, those who make an assessment, they have to decide like whether it is good enough or it's not, whether it will become dangerous. And sometimes you would, scientific people are sometimes can be biased because you are working on something passionately or there is an inherent interest, conflict of interest, and you want to succeed. And there has to be independent people who have to evaluate. Sorry. So the development team will include people, and it is usually a multidisciplinary team. We'll see that later. And they will decide whether a product is too risky to be brought to the market. And that's a very key goal, actually. Rejecting something, because a lot of effort, a lot of cost, there will be a lot of reluctance, a lot of defense will be there. And sometimes bluntly, sometimes politely, sometimes through discussion, you have to explain and halt, delay, refine, or stop the development. So for success, also it requires a multidisciplinary team, which will eventually decide at each step of development whether the risk is less than the benefit, or vice versa, whether the benefit is larger than the risk. And this whole team will decide. And the team will be guided by a team that knows about laws, regulations, and therefore the chances of further failure or success. Now, what is a good regulatory practice? I'll skip this slide. I'll come to this slide a little bit. I'll take this slide first. So when you see the team that are involved, like when is the regulatory involved in the product life cycle? Basically, like as you see the arrow, the blue arrow down, it's throughout the life cycle. So initial research and target laboratory synthesis. So a feedback has to come from people who are aware of the regulations and the SOPs, the quality manuals are based on regulatory guidances and laws. So whether it's a analytical lab, formulation lab, quality assurance lab, or CMC like the medicinal chemistry lab or microbiology lab, each one would have its SOPs and they will have a protocol for each method, each step, and the protocols will be designed as per the certain like given laws and guidances. So in this slide, you'll see that regulatory is responsible for internal review right in the early stage, the discovery stage, the preclinical animal tox stage, the human clinical trial stage. And it's very important in the CMC, which is the chemistry, manufacturing, and control. So formulations, chemistry, formulations, and even the control, like quality control of the product. So these are the areas in which you require the review by regulatory, the knowledge of regulatory, and people from regulatory who should be involved. what is a so the practice of good regulations should be inherently built in any research facility, whether it is a commercial organization such as pharmaceutical or medical device industry. Sometimes like the academicians have been a little away from regulations. The liberty of scientific mind, as it is called. I agree with that, but tend to now, not now, but last 15 years, I tend to disagree after seeing a lot of failures and questions, even in the industry when you start like the work in some of the Indian big companies where I have worked, Dr. Reddy's in the R&D of Reddy's, Kedla and Torrent Research Center. So even the pyramids here. So first 7 years was spent in the Indian companies, the big ones. And you understand, later on you understand, when you start working in MNCs, you understand that why certain things are failing. And gradually, like by the 5th year, I could, as a medical person, I was unaware. I'd not heard about the word SOP when I joined the industry, to be honest, 2001. And second year, I heard the word IQ, OQ, PQ, etc. So, and by the first year, I was already validating some of the instruments in my PK. Fortunately or unfortunately, I was, because of lack of a senior guy, I was heading the PK in Piramal in 2000, end of 2000, for the next 2.5 years. So the good regulatory practices have to be firmly established. And I'd say that the idea that regulations will come in the end and we will evaluate is not simply acceptable, whether it's the academic research or hospital-based research. Even like my colleagues in the hospitals, they will come to the regulators at the end and will say that, hey, it's already spoiled. Or in the academia, you come with a lot of like work and you evaluate it. And sometimes like you like appreciate the ideas. And it'll be a little bit of a reluctance to say bluntly, but hey, was the good practices followed? Good laboratory practice, good GMP practices, good GCP practices, good distribution practices related to supply chain, refrigeration, etc. So these, they fall under different heads, but finally, like the regulators have to read that and guide people that, hey, there are certain laws that you need to follow, certain guidelines that you need to follow at every stage. So although the freedom of scientific person, and we say that it's important to have an independent mind and like innovative mind, but unless you have certain abiding by certain like regulations, the risk is quite high. And perhaps that is one of the reasons, like not many products, they have come from the R&D pipelines of Indian academia, or which has been translated into serious human studies, or taken up by industry. And even the industry has failed actually because of huge time, cost, resource required in doing everything during the life cycle of 8 to 10 years with a high risk of failure, with complete compliance to regulations. But I'll say that it's not an option. It's not an option. So the earlier we learn that the good regulatory practice is critical function, as critical as making of formulations, uh, somebody synthesizing a medical product or polymer or a nano emulsions or suspension or something. So it is as important as anybody who's planning to do a clinical, like research on human or preclinical work on animal. You cannot just develop a protocol and run it without being within the framework of certain guidelines and regulations. So GRP is the practice of compliance with regulations and technology related to the product. The key word is compliance with regulations. So somebody has to know what is the regulation and ensure that compliance to regulations is followed in the technology and product that you are developing. Manufacture and control of the product. So CMC must be following the regulations. So for chemistry, formulations, pre-formulations, formulations, analyticals, the microbiology should follow the regulations, the toxicological and PK studies in animals, and efficacy and safety in animals and human must follow the good regulations. The good regulation practices will help in maintaining accurate information, which will help in product licensing. The chances of licensing will be higher. The principles of good regulatory practices are independence, accountability, transparency, and confidentiality. Now, what are they? Like, you should be independent. So normally there are three departments which we always feel and be independent of R&D. And by independence, you mean that reporting structure. So if you report to a person who has a conflict of interest in the product development, say the R&D operations head or the production and manufacturing operation head, he will have a conflict of going to the next step at all cost. And a production person will have the same idea, like because a failure might be a waste of a huge resource. So the quality assurance, suppose it fails certain substances and you have to discard it, or you have to recall a product which is already in the market. So somebody like should be independent to take a decision. So here, like regulatory, quality assurance, and drug safety team. We call it sometimes like pharmacovigilance, and sometimes interchangeably is the safety team. So these three teams which manage the risk and benefit should be independent. Regulatory, quality, and product safety of pharmacovigilance. They should be accountable for their decision because it's a huge decision. You are likely to take the product to the next step, or you are likely to fail it. And it used to be a huge challenge in most companies, particularly I'll say that Indian companies, because the resource is limited and there is a very compelling commercial decision to move on. So there will be a lot of pressure to go to the next stage to carry out the animal experimentations or even bioavailability or a bioequivalence study. When the analytical profile may not be completely satisfactory. And as a regulator, you have to put your foot down. And it's a huge cost, like it's a huge accountability because you are stopping a project from going further. So you should be completely aware of the legislation, directive, guidelines under which it does not follow, and why you are going to recommend review or stopping or the development, or go back to the next stage and redo the experiment because this is not acceptable to me. So huge accountability, and you should be transparent about your decisions. So both the internal regulator and the external. So the DCGI, the Drugs Controller General of India, in some of the not developed countries, like the regulators will have less knowledge. So the more knowledge you have, the better transparency you can have. And I'll give you an example as a medical professional. So when we explain a disease to a patient, if I draw the diagram of the eye or a GI system and explain that this is the antrum and this is the gut, this is the pylorus, and here the acid is synthesized and it goes to the upper GI, the esophagus, and it causes burn. So sometimes it is easier. And sometimes vaguely you'll see that it happens. It happens because of many things. So that is lack of transparency. And knowledge brings either the motivation to share information so that the other party is like capable of understanding the cause, and therefore they can go back to the laboratory and adopt a method that will improve the chances of success. So lack of transparency for both the QA and the regulatory team will damage the like the prospects of reverting a failure, or even like why a certain product was approved. So when you see the scientific review meeting of the DCGI, and I analyzed almost 2,700 of them from 2015 to 2017, entering each of them in an Excel sheet. And it was like questionable. You can go to the court of law and question them, and DCGI would be in serious problem. But since I want to maintain a good relationship, I can't go to the court for almost 100 or 200 bad decisions by the government agency. Confidentiality, it applies to everyone, particularly the regulators, because you'll be involved throughout the study. So you'll be like throughout the development. So you'll be privy to every data, and you'll be required to compile the dossier. We'll come to that later. So you have access to almost every, like from the birth to the death of a product in the company, very, very critical. Regulators. From here onwards, I'll move quickly. So unless there is, of course, like a 5 or 10 minutes extension, just like having a look at the watch. So regulators, this is a list of external regulators. I call it the health regulatory agencies. And this is a screenshot of my bookmarks. If you see the folder of the bookmark, Health Authority, and I have deliberately highlighted the EU. And in each of the country, like EU, like Britain is moving out. So the MHRA has already moved out of after the Brexit. It is in the transition phase, but they have already removed it. So the other countries, so similarly, like I have for almost 100 countries, the websites, and you keep looking at them. Although I'm not a complete regulatory, I'm more of a development guy. So regulatory is one of my like favorite areas. So having a tab of all the regulatory bodies, right from Africa to Latin America to Europe to Japan to India, like South Asian countries, because you don't know what is happening elsewhere, and if you don't know the regulations. So if you are working in a particular region or a country, you should know, and you should check that almost every alternate day or at least weekly for an update. What about the internal regulators? What are the necessary skills? We'll see that in the slides there, the subsequent slides, they are described. I'll skip this one. We have already dealt with this slide. So what are the necessary skills? Knowing the directives, legislations, guidelines, laws, and recent developments. So you need to check the websites for news or any updates, which will be on the landing page. Usually news and updates will be on the landing page. Landing page is the first page of a website. Necessary skill is like also required to participate in early stages of development. So most regulators, and that is because of the wide spectrum of knowledge that is required. Nowadays, the regulators are divided into different areas. We'll see that later. So somebody with early development or preclinical toxicology, and somebody with chemistry, manufacturing, and control background, somebody with a clinical background. So it might be difficult for a person, and for if you go to the FDA or the EU, European Union, there will be almost like 400 or 500 guidelines under efficacy, clinical efficacy, safety, preclinical safety, efficacy, toxicology, pharmacology, kinetics, etc. And there would be chemistry and control, almost like 100 guidelines. So somebody should, apart from the guidelines and law, there will be several other guidance documents and associated like texts, and people have to be aware. So it's very difficult to be familiar with everything. So in a typical large company, you would have regulations in the early development with the knowledge of early development, preclinical, animal talks. So a regulatory will be sitting within each of the team. So one of the job for a regulatory team is to make sure that the other functions and your colleagues in other departments are familiar and aware of the laws and regulations and guidances. And it should be participative, not objective or critic, but it should be a cooperative work as part of the team. But you should have the independence, but you should be part of the as a team. One of the key areas is the focus, like the ability of a person to compile, to understand data, whether it's preclinical data, chemistry or manufacturing data or clinical data, to understand the data, and then write the data into reports or texts that will go into a dossier and which will be submitted to the regulatory agencies for review, evaluation, approval or disapproval. Now, this is many of you must be familiar, like common technical document. So India also has adopted a common technical document, basically 5 modules. The module 1, this is a format that is followed for most products, whether it's chemical, biologicals or medical devices. The pattern will be slightly different, but if you follow like for pharmaceuticals, essentially you get a grip of everything. So first, module 1 is the administrative, like who are you, what is the company, where is the registration office, people, etc. Table of content, and then there will be 3 modules, basically broad modules: quality, which is the chemistry, manufacturing and control; module 4, which is the non-clinical studies, in vitro and in vivo studies, non-human. Module 5 is the human studies, phase 1, phase 2, phase 3, phase 4. Now, this is the way the documents are arranged. It is from my files. So this is the organization of the CTD. So this is the module 3. So when you see the top of there, it's a folder. So you need to have a folder called module 1, module 3, module 4, module 5. And when you look at the folder like 3.2P1, so it's related to the pharmaceutical product, which is the finished product. And it is the section 3.2 of product, 0.1 of the product. So similarly, it will be divided into, say, description and composition and so on. So right from the excipient, manufacturing process, reference standard, the container, the packaging systems, the stability data, the validation processes, etc. And when you open the folder, each one will have again the Word document, which will be like having certain like patterns. So 3.2P, 3.1, and so on. So they will be in a sequence. They will be all hyperlinked, so that you can jump from one section to the another, and that is called the eCTD, the electronic CTD. This is how they are organized. So it's a very minute, and they will be running into several thousands and thousands and pages, few lakhs total. I'm just opening one of the files, which is a short document, a plant master file for a medical device. So this is the Word document, the template Word document, which I had created for one of the companies. So basically to communicate about the business, it's a dummy. So the plant master file is basically like about the facility design and how do you comply with the good manufacturing practices. This will be included in the dossier, and the regulators have to be comparing with the legislation and guidelines whether it is compiled in a manner that it is okay. This is a summary of the technical documents for a medical device. So this is a summary, not the entire. So this will be module 2 of for a device, which is basically a summary. For chemical drugs and biologicals, it's huge, basically, about usually about 300 to 500 pages. So this is an important slide, and it's there on my website, and but it should not be taken as a conflict. So certain texts are removed so that any business related things are not taken out here. So when a product is developed for new geography, and the regulatory support, which could be internal support, so within the academic side or within the pharmaceutical, or whether it's a CRO or a consultant or etc. So somebody who knows the regulatory strategy. So the boxes in the light blue colors, they will be the broad departments which are available. So product regulatory strategy, regulatory documentations. So strategy is a guy who keeps reading, and commercial. So regulatory strategy is a new domain. Regulatory documentation, the guy who prepares the CTD gets the document, and it includes the CMC, and it includes medical team, CMC team, it includes the preclinical team. Regulatory archival labeling is what you call, if you have seen the summary of product characteristics, product monograph, many of you may have read or seen a Martindale or the physician's desk reference, or Mosby's drug reference. They are like what you call is like compilation of the labelings. So the draft artwork, which is the labeling, the inner labeling, the outer labeling, they are called artwork. Archival, now it is compulsory. So archival, like e-archival, so the physical archival, and now the e-archival, so the traceability, the documentation, the timeline for which it is required by law to be maintained easily. It should be easily available to the team, as well as to continuously update and reload the documents. There are different softwares available for that. Post-approval changes. So the job of regulators doesn't, when we say a life cycle, the job of regulators doesn't stop at the end of marketing a product launch. So after the approval, many processes will change. You have changed the API, the active ingredient source. It was a Chinese, now you are getting an Indian one, or you are getting a Japanese one. You need to resubmit the manufacturing data. You need to submit the stability data. You need to submit the formulation data with the new API compatibility studies, etc. Then comes the pharmacovigilance. So the pharmacovigilance I have included here in regulatory, but it has become a completely separate department now. But within pharmacovigilance, there is a group called regulatory. So vice versa, it is interchangeable. So regulatory can have a pharmacovigilance safety team, and pharmacovigilance can have a regulatory team. And their job is like basically pharmacovigilance will be more operational, and regulatory will be more like compliance to the regulatory guidelines. So I will, as a regulator, I'll be taking the documents from the pharmacovigilance team, what you call the individual case reports, or periodic safety update reports, or aggregate reports, and I'll be responsible for seeing whether it complies to the regulatory standards, and then submitting it to the regulators, and then coming back to the pharmacovigilance team that it is submitted to the regulatory. So I think this is one of the last slides. So product, where is your position, like for the regulatory team? And I say that's critical. It's right at the center, and it's along with the QA, I think, but it has a little larger scope because of its interaction and being being internal stakeholder, which is connected to the external stakeholder, which is the health agencies. So usually the regulatory team is the single point contact or liaising team with the external regulatory agencies. So either we have a web meeting with the regulatory agencies. So in multinational company, we used to support, say, my team in Canada or China and their regulatory agencies. So I'll be supporting them in joining a conference call, and then helping them in product registration in different countries. So a defense of, say, some like psychotropic substance with the TGA in Australia with the team there. So or IND, like initial development. So you will be having a investigational product discussion with the regulatory. You should be thorough with the documents, the content, have the internal discussion, because you are going to present it. You may carry with you other colleagues and you can pass it on to them, but basically you are the point of contact, and therefore the comprehension and knowledge of different work is important. And as you go up the ladder, like when you become like a therapeutic area head in the regulations, then you should have the entire knowledge. Like gradually you pick up everything that is possible in a product development. And generally, I'll say that for a QA and for a regulatory, it is somebody who is not greenhorn, somebody who is new and you groom them. It's difficult. And I would always recommend rotation. So a person who rotates through different, say, he has spent 1 or 2 years in formulation, 1 or 2 years in preclinical department, microbiology, and then you come and become a regulator that's much more competent than a person who joins immediately and he remains within a particular department throughout his life, because sometimes it's difficult to correlate them as a senior management in regulatory. So at a junior level, it's okay, but as a senior level, unless you have experienced a flavor of everything in drug discovery and development, it is almost impossible to head a regulatory department. Next slide is the last one. Goal of the regulatory is to keep it safe and effective. So this is the last slide. The ultimate job, whether it's an internal regulator or external regulator, the ultimate job is to ensure that the product that goes to the system, to society for use in human are safe and effective. And sometimes like cost is also nowadays included. So now regulatory, they also need to be aware of a little bit about cost, cost and quality of life. So a little bit of that is added to ensure that the word that is being used more often, the value. So the value is equal to safety on one side, and the cost, effectiveness, and the quality of life. So with this, I will end the slide, the presentation, the main presentation, and now like we'll have a little bit of a short, I think, time, but it will be good enough, I think, to take as many questions as possible.
Thank you, sir. Now it's the session for the question and answers. So here are some set of few questions asked by Aditya sir. The first question, it is safe to have a fast track discovery or development of any new medications or formulations for any sort of emergency?
There is nothing like there is a fast track development, and there are certain rule, for example, for COVID or certain emergency situation, certain guidelines are quickly developed and passed, but you cannot have your own this thing, own ways to start doing something unless, and for that you need to check the regulatory websites of the country. Suppose you are developing for India or you are developing for a particular target. So you need to check. Suppose you are developing something for COVID for USA, might be different from India. And there will be several shortcuts in certain preclinical work, the sample size, like the p-value, like, okay, the power of the studies, certain exemptions might be there, but still you need to check what is available in the guidelines, because if it is not updated in the regulatory websites and guidance, you have no option, and you have to still follow the old, like the traditional details.
And the second question, are side effects or ill effects one of the most important factor which is determines the new drug or medication?
Yeah, so the question here, madam?
So they have asked, are side effects or ill effects one of the most important factor which is determines the new drug or a medication?
Yeah, basically like one of the factors and that is the job both in the preclinical safety and now the certain simulations and analytical predictions models can be used even before the in vivo studies. So basically there will be toxicology, and when it comes to a human we call it safety. So because of toxicological side effects, which could be it could be renal, hepatic, gastrointestinal, dermal, various toxicities, blood and hemopoietic system. So almost but when you look at the lead discovery, candidates, in the older days, more product used to fail because of the PK. Gradually, more products they fail because of toxicity, and more as you go through the preclinical stages and animal tox stage and early development, human phase 1. There are chances that it will fail because of the undesirable side effects in human or toxicological profile in animals. Say even a single animal, it shows say a fatty liver change. In 5 animals it is not showing that change, and you present a data of a mean of 5 animals. As a regulator, I would want individual data, and if you cannot explain why one animal had a fatty liver, I'll hold your product. It will be on hold because I need an answer. Because one have a fatty liver in out of 6, 15%. What will happen to the next stage if it is given to a human? And it will not be acceptable to me unless at least you have a theoretical answer to explain the cause of fatty liver in that animal. So yes, safety is one of the big reasons why product will fail in the development, discovery, and development stage. Most products they fail in the preclinical animal tox, and in the acute tox, and in the those which succeeds in the acute tox. Many of them they will succeed in the chronic tox. It gives a good idea. But the 30 days, the idea of acute tox is becoming obsolete because most drugs are not given a single dose. Europe has removed it. So in 30 days tox, many drugs will fail in the 30 days tox because there will be some changes. Say the hemoglobin has come down from 12 point something in animals, like 13 or something to, or 10 to like 10% decrease, and you are not able to explain it. It will not move further. So it becomes important, like any of the parameters, if it, there are still like different textbooks for animal pathology and biochemistry data. And if it doesn't fit within that range, and if it is out of range for whatever reasons, it will be very hard to take it to the next stage. And in human studies, most of them they will fail in the early. So more drugs they fail because of efficacy in later stages. Stage 2B and 3 they will fail because of efficacy and safety efficacy ratio. But more drugs will fail because of safety in early stages, PK and safety in early stages.
And the last question, sir, with increases in the resistance of microbes like coronavirus from many factors, does the medication or vaccine needs changes, though it should be high success or low risk in past? I'll repeat, with increases in resistance of microbes like coronavirus from many factors, does the medication or vaccines needs changes, though it showed high success and low risk in past?
Madam, if I've understood the question, like, is it the existing vaccine, the use of the research on existing vaccine
Yes,
to
sir.
try it out on a COVID, for example?
Yes, sir. Yes, sir.
So it becomes a new indication, right? Like
Yes,
the drug is same, its chemical and pharmaceutical property has been established. Now it becomes a new indication that I want to try. So here you don't have to do a phase 1 studies because it's already there in human beings. So certain data of in vitro data that it is effective in, say, in COVID, there is a claim that there is a resistance. So theoretically, literature and other things might support, but not acceptable. So whether with that vaccine, any IC50 values of in vitro test tissue culture, I'm not completely competent in this area, but I would guess. So IC values of like in vitro tissue cultures and whether it is able to inhibit and whether it's a passive immunity or the vaccine is for passive immunity or active immunity. So like if it is for say active immunization, but here like in COVID, we are looking at a passive immunization, which is like it develops and therefore prevents it. So in tetanus, it is an active immunization. Rabies, it is active immunization. After the disease is there, you give it. So here the case is about a passive immunization, and you will probably see the cells whether they are able to generate certain antibodies, which are biomarkers for in vitro studies. And say, as far as I know, like in case of COVID, like interleukin 6 and tumor necrosis factors and C-reactive protein in vitro, whether and whether the immune response is seen in tissue culture and cell cultures models. And animal models, like I'm not very sure whether it is a good idea to like experiment on animal models. Here is a case where there will be some exemption, and because there is a high benefit and high risk. So when we talk about disease like COVID and there is established medicine, for example, like hydroxychloroquine, it has a high risk, but it's worth trying. And I'll say that for me, it should be given actually. Although US is not yet, but many countries have recommended. So as a healthcare provider, as a regulator, my risk is high and my benefit is high. So the risk benefit ratios also changes because the benefit is also high, the risk is high. And because the benefit is larger, because how many patients will die if I don't give anything? Say serious patients who are there, this thing. Yes, if it is for prevention, unlike tetanus, where there is a great likelihood of dying in if you don't give like in certain detected tetanus cases. So here, like in prevention, the benefit becomes low. So if I say that yeah, I'm trying to give it for passive immunity and where the benefit is low, because if you give the vaccine, how many you actually get? So less than 1% are getting, if exposed, they are getting the infection, or 50% are getting the infection when exposed. But normal population, only 1% is getting the infection. So should I expose everyone to the vaccine? I don't have the data right now to recommend that. So what will be the internal regulator saying or the government agency is saying at this moment? Should I make it available to high-risk patients, say doctors and others who are uh, likely to be in contact now and in near future? And a limited clinical trial should be done to see like the response, whether it is able to like prevent in high risk exposure cases. So I would say that certain strategies can be planned to check the existing vaccines, but it will be difficult for a new vaccine because the pre-formulation work, the chemical compatibility, the formulation work, stability, etc., is a huge work, and the toxicological work in animal before it goes to human. So I'll be skeptical like of a shortcut in a new chemical entity or a biological entity. But if it's the existing one, you can jump to high risk groups and do a limited trial, a phase 3, a phase 2B, and a phase 3 with a small sample size. There are certain protocols for doing clinical trial in small sample size, and I think this would fit into that. So the answer would be like, again, it's up to the regulators. And unless they pass a law and certain guidelines and it is available, it will be not be judicious for somebody to do it. And as a physician, somebody might use a vaccine. Say, for example, I'm a physician, I use as the pneumonia vaccine, like influenza vaccine, to try and see whether it works in this thing. If it is a research, it needs an approval. But if it is a discretion of a medical practitioner, then the only individual liability is there. So this becomes more of a legal question. So if I'm doing a research, systematic data collection and its interpretation, it becomes a research. It requires ethical committee approval and drugs approval. And if I'm using it for a patient, individual patient in his best interest, I feel as a physician, this patient might die and I might use it. So that's the discretion, and it is guided by the law of like a medical practitioner, his privileges, and whether it amounts to, like, if anything happens, medical negligence or malpractice. It could lead to, like, say somebody, a case of human slaughter or gross malpractice or negligence. So in short, like, unless the external regulators, they define something, it will not be prudent to do something which is not already defined.
Thank you, sir. There is a question. COVID-19 vaccine regulation. How is it different from the traditional vaccine regulation?
It is not different. So the regulations are same for development of a vaccine. Particularly for COVID, because of health emergencies, certain documents have been released, which is like guidance documents for development of a vaccine. And basically, like, you will be following the same process because you cannot shortcut the quality control and manufacturing processes and try out, say, a phase 1 study in human being without enough safety and efficacy data. Here, some of the guidelines which, and I'm not 100% sure because I've not focused on the COVID like development and the vaccine development guidelines, but I know that they are being released. But essentially, like, they have to follow the basic vaccine guidelines. So immunogenicity, the chemical stability, and other like stability data they have to. Now, there will be exemptions on stability data. Instead of 6 months, you can generate a stability data of, say, 3 months or 1 month and extrapolate it. Instead of 2 animal sets, you can do for 1 animal, and then the skin tests and proceed whether it is local toxicity is there or not. So these are roughly, I'll say, without going into detail of the guidelines, I'll say that these are the things that would be or have been recommended. I'll say one needs to go back and check the regulatory guidance, particularly with respect to COVID. But essentially the formula will be same. And there will be certain stages will be like limited, like the requirement of 2 animals and 2 routes of toxicological studies, stability data for 6 months, the requirement for a large study which meets like 80% power. So it could be a pilot studies, and after a pilot study, you immediately go to a pivotal study. And in USA, for example, for a product approval, you require 2 randomized trials. Here you can like have 1 randomized trials, and the need for a comparative trial will be waived off. So I've not seen the guidelines, but this is my guess actually, and I'm sure like it will not be outside this. It will be too dangerous to go outside this.
So there is one more question. If the medications show any, presently if it's not showing any ill effect, but later on if it's showing any effects, it should be withdrawn from the market?
There are good cases. If you know like terfenadine and yeah, the last one like celecoxib, like many of us remember celecoxib, like rosuvastatin, um, which were withdrawn in certain markets. So there are drugs that, particularly the coxib was a remarkable one, and certain like antifungal drugs, antihistamines, which were withdrawn. So terfenadine is a typical one, and it was withdrawn after 14 years of use. And celecoxib, after it reached almost few billions in sales, they were withdrawn. So it's a difficult decision, and a lot of debate has gone in, and whether the pharmaceutical companies, they don't present complete data, and therefore the need for a robust pharmacovigilance data, independent pharmacovigilance data. So the government agency, for example, in Europe, because earlier like the pharmacovigilance responsibility was with the company entirely. So I may present, I may not present, and I will be audited for my pharmacovigilance. But you need to have independent pharmacovigilance like feedback. So there are drugs which were approved and then withdrawn after several years, even after 10, 15 years of being in the market, they were withdrawn. It shows that the complete information is not available. By the time you complete your phase 3 studies, say in approximately 3,000 or 5,000 patients, like anything roughly about say 3 or 5,000 patients, an adverse event which has a occurrence of less than 1%, it is very difficult to detect that in a large number of, to be very sure. So something which is like genetic based, certain only the people with genetic predisposition, or certain genetic, like people who have certain CYP chromosomal profile, so they might experience the toxicity. But in a clinical trial, I'm not going to do a segregation and sub-stratification and follow up different groups for certain genotypic traits. So certain diseases, certain patients will be very difficult unless there is a large number of patients are exposed. So in a post-marketing setup, unless several lakhs, millions of patients are exposed, and that is where the pharmacovigilance drug safety comes in. And the risk minimization strategies, the emergency drug recall strategy. So you use the word recall, and there has to be an SOP and for a drug recall, like not one SOP, there are usually several SOPs. You discover that certain, you remember the nimesulide. So getafloxacin and like several quinolones, except like Cipro and others like sparfloxacin and others were also drugs which were withdrawn because of photo and hepatic toxicity. So here, like suppose in USA it has been withdrawn, and tomorrow the DCGI in the morning declares that all products and I have faced this situation in company. So at least twice. So gatifloxacin is withdrawn, and then they have not declared whether all formulations or only ophthalmic, and we are dealing with only ophthalmic. So the drug inspectors throughout India, they have gone and started confiscating our ophthalmic product. And then I have to coordinate both with the DCI. If you remember, like recall Dr. Y.K. Gupta and others, they were in the committee. So then you have to negotiate both with the DCGI people as well as the state drugs controllers and the inspectors. Like, hey, we'll go to the court if you this thing, because the guidelines which is issued is not very clear, and I'm not going to recall the product. Whereas like those which have systemic injections of sparfloxacin or gatifloxacin, they were withdrawing it from the market. So you have to have the procedure for a recall, because by the time, suppose it's a acute, it's a contamination, or if it is a known side effects. So the side effects could be because of even a contamination, quality related. So entire batch goes out, and then it has happened quite a number of times, like the polyethylene glycol being contaminated with certain other excipients, toxic excipients, and mass casualties. And then these are adverse events, and you have to have a process for quick recall within 24 hours, 48 hours. If it is an emergency recall, right from at the patient level, retailer level, distributor level, CFA, stockist and clearing and forwarding agency level, whether you have a process in place to recall that product in case of emergency side effects. But some side effects are known, for example, like clozapine, olanzapine, or rosuvastatin, celecoxib. It is being discussed with the company for several months, and then you know that it's going to fall. The knife is going to fall, and then you start like you become hesitant in production. Say nimesulide. So companies will have an internal discussion. I was in Dr. Reddy's when the nimesulide thing came up. So emergency meetings and other things like we'll generate data, we'll defend it. We went on to defend in other populations, and they accepted that. Later on, there was some other guidelines for adults. It can be given, but pediatric it cannot be given. I think 2014 or 15. So because of the adverse side effects, not in Indian data, but this came from the French data. So it could like it could happen because of known side effect in the native population, which is in India, through the pharmacovigilance data, and it is sometimes voluntary. The company detects it and voluntarily goes to the regulatory agency and withdraws the product. Sometimes the regulatory agencies, they detect the problem with the product and then they recall it. Sometimes that is a emergency situation because of certain quality problem, and then the products are withdrawn.
Thank you, sir. Any more questions? If there is no questions, now we came up to an end of the session. At this moment, I like to thank our management for their constant supports, and I like to thank our beloved principal sir for his constant support and the encouragement. And I like to thank our IT department for their technical supports. Last but not the least, I like to thank all our delegates and my dear students who has given a constant support as well as the encouragement for this grand success. Not our colleagues who has given the guidance and support to be a successful of this program. Thank you one and all. A kind notice to all the delegates, the feedback link will be sent to your registered mail ID. As soon as you have submitted your feedback, the certificates will be generated to your registered mail IDs. I'll repeat. For your registered mail ID, the feedback link will be sent. And if you have submitted the feedback, the certificate will be automatically generated. Thank you, one and all.
Thank you, everyone.
Thank you, sir.
Yes, it's fine.